we're having, so we've switched up the MBZ retreat this year, and this was due to discussions that we had at the last retreat. A lot of people saying, well, this isn't what we were having, wasn't really a retreat, it was a symposium. And what we'd like is a more social retreat. So we're having that, and Lelania sent out various emails about that. The time when we've normally had the retreat in the past, which has been on Wednesday of RRR week in spring semester, we're now going to have a genuine retreat, I guess, at Hastings. So that's going to be a one overnight going down on Wednesday and coming back on Thursday. And you'll hear more from Lelania about that. But this was in response to demand from everyone, both students and faculty and postdocs alike. So I really hope people attend and participate. I think it'll be really fun. So that's mostly going to be a social affair. But then what I've always called the retreat, but I guess now we're going to call the symposium, it's still going to continue. That's an MBZ tradition that we're going to continue with, where everybody gives a talk. But this year it's going to be, Lelania, what's the date?
It will be on Thursday the 17th of January.
So right before classes start.
Right before classes start, and it'll be at the UC Botanical Gardens. So
rather than being in the Brazil room at Tilden, where we've always done it in the past, it's going to be in the Botanical Gardens in a similar kind of room. A one-day thing, close to campus. Everybody is expected to attend. The MBZ will be closed that day. So I just want to make sure that's on everyone's calendars, because that's coming up before the next MBZ seminar. Okay. The other announcement I have is that this is officially a class, and 18 of you are enrolled. It's a class that's co-taught by Rory, Jim, Eileen, and me. And so we need you to fill out teaching evaluations. So after, sorry, the university asks for this. So after the seminar today, please fill out 4 of these, one for each of us. Sorry. We're required to ask you for that.
Don't they need that as a duplicate?
All right. Any other announcements? Okay, it's
You have
really
one more announcement.
one more announcement.
You have one more announcement.
That's coming.
Okay.
All right. It's a real pleasure today to introduce Katya Mack, who's giving her finishing talk. Every time a student graduates, it's a big deal, and for me personally, this is a very big deal. I've really enjoyed having Katya in the lab for the last 5.5 years. She got her undergraduate degree at the University of Michigan, and she worked with Milford Wolpoff and Tricia Whitcop. She's been here at Berkeley 5.5 years, and after she leaves, she has a postdoc lined up at Stanford with Hunter Fraser. She won a competitive Stanford Genomics Fellowship to go there and continue work on gene expression in yeast. You know, Katya, I knew early on was destined for success, and I knew it when I read the first sentence of her application to graduate school, which I will now quote. This is the first sentence of her application to graduate school. When I was 7 years old, my father gave me a Neanderthal kit, complete with a miniature Neanderthal skeleton and booklet. For at least the next year, I was rarely photographed without a book on evolution in my hands and my Neanderthal skeleton clutched to my side. While this gift did not make me an instant hit in 2nd grade, it did spark a lifelong fascination with biology and evolution. So after I read those sentences, I knew right then and there I was going to accept her to graduate school. She went on to say in her application, I am particularly interested in mammalian gene expression evolution, especially as to how genetic interactions affect the speciation process. And you know, it's remarkable. Not everybody goes on to do exactly what they say they're interested in, but Katya has. Her interests have really focused on gene expression evolution in mammals, and she's been enormously productive in this area. She works very hard, she's smart. She's really what I see as a model graduate student. Her thesis has 5 chapters, 3 of which have already been published, 1 in Trends in Genetics and 2 in Genome Research. Each of these is a major contribution in its own right. The other 2 chapters of her thesis are written up and ready to submit for publication. So she's going to leave here with her thesis either published or submitted. In addition to that, she has a number of side projects that she's contributed an enormous amount to. We have this gigantic project with mice in enclosures, an experimental hybrid zone project that Katya started and put in many months of work on. So she's really contributed an enormous amount, and then there's several other big projects that are still underway that will lead to separate papers. In addition to all of her science, she's been a great teacher. She's taught every semester she's been here. Is that?
Except one.
Except one, when you got an MBZ fellowship. Yay, MBZ. And despite that very heavy teaching load, she's really been exceptionally productive. She also has worked in this Be a Scientist mentor program in public schools. She's regularly taught workshops on RNA-seq for the CGRL. She even gave a short course at the Cold Spring Harbor. Nipan Patel organized and asked her to teach him. So she has this great contribution to teaching as well. I personally have tremendously enjoyed all of our intellectual interactions over the years. When I have an idea I'm trying to work out, I often go to Katya. She's been a great sounding board. She's been a source of ideas. I remember in particular sparring with you over this idea that we could look at gene expression in natural populations and identify cis-EQTL in these mice that we had sampled. I had just come back from a visit to Duke University, and where I chatted with Jenny Tung, who you've all invited to speak here next semester, and she had just published a paper on this, and I was very impressed with this, and I suggested to Katya, well, maybe this is something we could do in mice. She had done it in primates, but we'd sparred about this for several weeks, and it really was an example of the kind of back and forth that I found particularly enlightening and helpful. She's helped me as I've worked on grants. She's been a great sounding board for that. So in all respects, she's an excellent scientist. Now, you know, everybody has their other quirks, and Katya is no exception. So I was trying to think, you know, what anecdotes can I tell? Well, you know, when you think of Katya, you think of smart and hardworking and all of that. But she also is just maybe a little bit high-strung. I guess that goes with this competitive, hard-working thing. And so you've probably noticed, if you've gone to visit her in her office, that most of the time she's focused like a laser in front of her computer, and it's as if the rest of the world doesn't exist. She's right there. And I've realized over the years, when I would walk into the office to talk to her, the reaction I usually got was a jump about 3 feet off of her chair. She I was always surprised. So I got in the habit of, when I wanted to go to talk to her, to walking in very quietly and making a big circle around her to sort of come into her field of view and not surprise her. And then she would look up and be a little less jumpy. I think that's just an indication of how focused she is on her work. The last thing I want to say is that Katya has been generous beyond measure. She has contributed to the lab in so many ways. I'm sort of a Luddite when it comes to things like developing web pages. Without asking me, Katya just took it upon herself and built the Nachman Lab web page and said, all you have to do is push this button to activate it. And that's just one of many, many examples of her generosity. She's helped everybody in the lab in various ways, and many people in the museum with computational issues. So we're all very grateful for that. So I just want to say publicly how much I appreciate all you've done for the lab and the MBZ, and how much I've enjoyed having you with us. This afternoon we're going to celebrate with a cake and some bubbly at 3 p.m. in here. Everybody's welcome to join us. I've already taken too much of your time, so with that I'll turn it over to Katya and say congratulations for 5 great years in the MBZ. So take it away.
Thank you so much, Michael. That was really, really nice and unwarranted. Today I'm going to be talking to you about a role for gene regulation in house mouse speciation adaptation. So nearly 40 years ago, Mary Claire King and Alan Wilson famously described a similarity in proteins between humans and chimpanzees, and found that the degree of molecular similarity you see, they thought couldn't explain the vast anatomical and behavioral differences you see between the species. They proposed that perhaps changes in gene regulation frequently were a big contributor to differences between species. And in the time since then, there's been a lot of progress in understanding the mechanistic basis of gene regulation, as well as characterizing differences between species, but there's been less, and the questions remain on the role of gene regulation in evolutionary processes like adaptation and speciation. So today I'm going to talk to you about two specific projects from my thesis. In one, I look at gene regulation and speciation in house mice using Mus musculus and Mus domesticus. And in the other, I look at gene regulation and environmental adaptation in a Cline from North America. All right, so starting with this first part, the genetic basis of post-zygotic isolation is of great interest for studying the formation of new species, and until recently, a lot of the work in this area has focused on identifying speciation genes. So those are specific genes that are involved in speciation. And until recently, there's been a lot less focus on gene regulation in this process. And this is a little surprising because gene expression necessitates the interaction between loci. And a lot of models of hybrid incompatibilities also require the interactions between loci. So that led us to the question of do changes in gene regulation also contribute to speciation? And lending indirect support to this idea, a number of studies in the last 15 years have found that hybrids between species will often have unusual expression patterns. This is called misexpression, and it's when the expression in a hybrid falls outside of the range in what you see in the parents. And this is very common. So just to tell you a little bit of, or remind you a little bit about how gene regulation works on the transcriptional level. On the transcriptional level, you have cis and trans regulatory elements. Cis-regulatory elements are stretches of non-coding DNA. They bind transacting factors, which are frequently transcription factors, to regulate the amount of mRNA that's produced. And changes in gene regulation have been found to be frequently deleterious. And so there's a lot of evidence now that gene expression itself is under stabilizing selection. So work in mutation accumulation lines, as well as in comparisons between species, have found that frequently there's stabilizing selection acting on gene expression levels. So, as cis and trans elements need to interact harmoniously to produce an optimal amount of mRNA, you could envision a model in which divergence between them could contribute to hybrid incompatibilities. So, if we imagine that we have this ancestral population where there's a transcription factor that binds to a cis-regulatory region to produce a certain amount of mRNA, and then these populations diverge, or this population diverges. And then you might, in one population, have a mutation in a transcription factor that changes how it looks, or how it binds. And in the other population, you have a change to the cis-regulatory region. In these isolated populations, that's actually not an issue in this example. So this transcription factor that is changed, as indicated by the star, is still binding to the cis-regulatory region. And this cis-regulatory region with this change is still binding this transcription factor. But in hybrids, you can have situations where those, this transcription factor and this cis-regulatory region are no longer binding correctly. And so then you get misexpression. And so you could envision this happening across many, many genes and then resulting in hybrid dysfunction. Now, if gene expression is frequently under stabilizing selection, you could also imagine that this might happen in a single lineage where you have a mutation in a cis-regulatory region and is compensated for by a change in a transacting factor. And then you have the same issue in hybrids. And these models that I just showed you are consistent with the Jansky-Mueller model, which is the common model for hybrid incompatibilities. So the other thing is that this model that I've just shown you makes a really explicit prediction, and the prediction is that for misexpressed genes, we'd expect them to be associated with changes in cis and trans together rather than saying changes in cis or trans by themselves. So to test the key predictions of this model, we used the model system house mice. House mice are a great system for asking questions like this because Mus musculus domesticus and Mus musculus musculus, which for the rest of the talk I'll just call musculus and domesticus, are recently reproductively isolated. They diverged around 350,000 years ago, and they form a stable hybrid zone in Europe. And they're partially reproductively isolated in that you can produce sterile and fertile hybrids depending on the genotype of the parents. So today for this project I'm going to go through, we're going to talk about first expression divergence between musculus and domesticus broadly, what the contribution of cis versus trans variants is to the divergence between musculus and domesticus, and then the role of selection on gene expression levels in musculus and domesticus. And then I'm going to talk to you about misexpression and hybrid sterility. So we're going to look at whether misexpression is associated with hybrid sterility, and then whether misexpressed genes are associated with opposing changes in cis and trans, which would be expected under the model that I just showed you. All right, so to do this, we used pure species of Mus musculus and Mus domesticus, as well as hybrids. A really great thing about these inbred lines we have in the lab from musculus and domesticus is that you can cross them in one direction, and you can take a musculus female and cross it with a domesticus male, and you can produce a sterile hybrid. And then you can also alternatively cross the domesticus female with a musculus male and produce a fertile hybrid. So you can actually directly contrast sterile and fertile hybrids and look at misexpression that's associated with hybridization versus misexpression that's associated with sterility. And so we had 3 samples per genotype of the parents, so the pure species, musculus and domesticus, as well as the F1 hybrids, the sterile and the fertile. All right, so to detect cis versus trans regulatory variants, what we do is this test devised by Patricia Whitcup 10 years ago, where you basically imagine a scenario where you have one species that's expressing gene A at a lower level than species 2. You're interested in whether that change is due to something acting in cis, so perhaps a change in a binding site, versus a change that's happening in trans, so perhaps a transcription factor itself has changed. So what you can do is compare the expression between those species and their hybrids. So in a hybrid, you have one of each allele that are now sitting in the same transacting environment. So any difference you see between these 2 alleles has to be due to a cis-regulatory change. Because again, those alleles are sitting in the same transacting environment, so you wouldn't be able to see differences that are a result of trans. Alternatively, if the expression level is the same in the hybrid, even though you see differences between the parents, that's good evidence that there's been a change in trans. So how we do this practically for the mice is we're looking at the number of reads that are mapping to the domesticus versus the musculus allele in a hybrid, and then what's mapping to the domesticus parent versus the musculus parent. And so again, for the cis-regulatory changes, those are going to fall along this axis, and this is just predicted data, this is not real data. Basically what we're seeing for under just a pure cis-regulatory change is the difference you see between the alleles in a hybrid are recapitulating the differences you see between the parents. Okay? So alternatively, for a trans change, this is a case where, again, you will see differences between the two, between the parental species, so they're falling away from the 0 on this axis, but you don't see differences between the alleles in the hybrid, so they're going to fall along here. Now there are, of course, cases where you have both cis and trans changes together, and so you are looking basically at how well this ratio is recapitulating this ratio, and I'll just point out one specific example because I'll talk about it a lot more later. But one more pattern we can see is compensatory changes, and those are cases where we don't see differences between the pure species. So musculus and domesticus are showing the same level of expression, but when we look at the hybrid, the hybrid is showing differences between those two alleles, and that suggests that there might be compensatory evolution. All right, so this is all predictions. Now I'm going to show you the real data, which looks like this. All right, so what you can see is that there's a lot of regulatory divergence. First of all, there's a large component of cis-regulatory divergence. About 24% of genes showed cis-regulatory divergence. A much smaller proportion showed changes in trans, so just 9% showed divergence due to trans alone. But the majority of cases we saw were cases where we had cis and trans acting together. And so, like I mentioned, there are cases where cis and trans are acting in opposition, so they're counteracting each other, versus cases where cis and trans are acting together towards expression. And based on that, we can actually then ask whether there's evidence of selection on expression levels. So looking at these 4 categories, and these are again the ones where cis and trans are acting together, we can ask whether, first, if under neutrality, our assumption is the number of cases where cis and trans are working in opposition to each other will be the same as the number where they work in the same direction. Under stabilizing selection, we'd expect more opposing changes than changes that are acting in the same direction. And then finally, under widespread positive selection, you might expect the opposite, where changes are always acting in the same direction, and so there are far more of those than what's happening in opposition. What we see is a very clear signal that's consistent with the previous literature, that there's a lot more, so here, to orient you, here's the expected number versus the observed numbers, and we're seeing a lot more observed opposing changes than what we would expect, which is consistent with widespread stabilizing selection on gene expression levels, which, like I said, is consistent with that literature on mutation accumulation lines. Okay. So now I'm going to move into talking about misexpression and hybrid sterility. So this is focusing in on looking at comparisons between the sterile and fertile hybrids. So the first thing we looked at was just how many misexpressed genes were found in the sterile versus the fertile hybrid. So the way we did this was just look at genes that exceeded a certain fold change compared to their parents, compared to both parents. And we see that there's a lot more, as indicated by the giant X, these sterile hybrids had a lot more misexpressed genes than the fertile hybrids. I'll talk more about this in a second, but a lot of these genes were actually also related to reproductive phenotypes, which is interesting. And then, given that number of misexpressed genes, we were interested then in testing our original question that I set up for you about whether misexpression was associated with specific regulatory categories. And so we asked whether that misexpression was associated with changes in cis and trans together and independent, or changes in cis and trans that were together and opposing. And what we see is that there's a very strong enrichment of cis and trans together and opposing with misexpression. So that supports that model I said to you at the beginning, but it's also interesting, those 29 genes themselves are interesting because these are all genes that have a cis UQTL, and so they potentially make good candidates for following up and studying hybrid sterility more closely. So I'm just going to show you a few of the genes in that circle, and so these are just a subset of them with annotated phenotypes. A lot of these have phenotypes related to expression in the testes and gonad development and spermatogenesis. But there are also a couple that I've starred here. So there are 5 that I've starred, which are particularly interesting to us because they appear in regions that have been found in 3 other studies. So those 5 genes are in regions that have been identified from a QTL mapping study on hybrid male sterility, a scan of the hybrid zone, so these are in regions that don't intergress across the hybrid zone, indicating they might be involved in hybrid sterility, and they're also in regions that are highly differentiated genome-wide. All right. So to sum up that first part, we found extensive evidence of regulatory divergence in cis and trans between Musculus and Domesticus. we found that there's evidence of widespread stabilizing selection acting on expression. And then we found that misexpression, there's more misexpression in sterile hybrids and is frequently related to reproductive phenotypes. And that misexpression was associated with opposing changes in cis and trans. And so just to remind you of that model that I showed you at the beginning, this result is consistent with this, and it's consistent more generally with the idea that regulatory interactions might contribute to the Jansky-Mueller incompatibilities and be important in the speciation process. Okay. So moving on to the part 2, which is changing gears a little bit, I'm going to talk to you now about gene regulation and environmental adaptation. So obviously understanding the genetic basis of adaptation is a major goal in evolutionary biology. And it's been cis-regulatory changes, so changes in those local binding sites, are predicted to play a disproportionately large role in adaptation. And the reason for this is that cis-regulatory changes, variants may be less pleiotropic than changes to proteins themselves. And to visualize that, you can think of a case where we have a gene that's associated with 2 specific cis-regulatory elements, one that's kidney specific and one that's liver specific. And those bind proteins that regulate the expression of this gene. So we have this transcription factor that binds in the liver that regulates the expression of this gene. And if you change that if you change that binding site in the kidney, that'll change the expression of the gene, but it wouldn't have changed the expression of the gene in the liver. However, if you were to change the protein itself, you would potentially change that gene in the liver, the kidney, and anything else that it was expressed in. So in this way, cis-regulatory variants can be less pleiotropic and can be spatially or temporally specific. So there's now some, there have been a couple clear cases where cis-regulatory variants have been linked to adaptation. Some of the more well-known examples are, for example, lactose tolerance in humans and color variation in Peromyscus. But something a lot of these examples have in common is that most examples involve genes of large effect, but most traits are polygenic and complex. And so we'd ideally like to be able to understand most traits. So a great example of a complex phenotype that's thought to be adaptive is latitudinal body mass variation in many species. So this is a geographic pattern known as Bergmann's rule, where you see a tendency for body size to increase with latitude, and this is thought to be a response to differences in temperature. And one way to visualize why this might be the case is if you think of a cube. As a cube increases in size, the volume of the cube increases twice as fast as the surface area. So larger animals have smaller surface area relative to their mass, and therefore would be better at conserving heat because they're going to lose less heat. So it's better to be a larger animal at a higher latitude. So like I said, this is an ecogeographical pattern that is well known across many species, including humans, but more importantly, it's also seen in house mice. So today I'll be talking to you about identifying evidence of adaptive regulatory evolution using house mice collected from a Cline by Megan Pfeiffer-Rixey in the east coast of the United States. House mice are actually recent additions to the Americas. They're human commensals, so they live in a lot of human disturbed structures, and they traveled from Europe with humans into the United States about 300 to 400 years ago. And on the east coast of North America, they show clinal patterns that are consistent with local adaptation, including variation in body mass. So mice from New York are much larger than mice in Florida, and this variation is clinal, so intermediate populations have intermediate body sizes. These differences persist in the lab, so what I'm showing you here, all the blue dots on the next couple slides are going to be New York, so you can think of New York blue cold, and red will be Florida. So even over a couple generations in the lab, New York mice are consistently larger than Florida mice. indicating again that this has a genetic basis. There's also differences in blood chemistry, including adiponectin levels, leptin levels, triglyceride and glucose levels. And finally, there are also behavioral differences in the lab. So mice from New York build bigger and more beautiful nests. And mice from Florida, and mice from New York also run faster. Or more, I'm sorry, they run more. So this leads us to the question of what the role of regulatory variation is in this environmental adaptation. And this is a project that involves Mallory Ballinger, who is on the right in that picture. So to do this, we used samples from 5 populations, 10 individuals per locality. Liver was collected in RNA later and used for gene expression. These mice were, by the way, again collected by Megan Pepper-Rixey. And then we also have exome sequence data for most of the individuals that we also have RNA-seq data for. So to identify adaptive regulatory variation, we were looking at the overlap between 3 different signals. First, we were looking at allele frequency Clines. So Clines and allele frequencies can be evidence of local adaptation when not confounded by demography. We then looked for cases where those alleles that were Clinal also were contributing to variation in expression, or they were expression quantitative trait loci, EQTLs. And then we were asking, do those overlap then? Are those EQTLs regulating genes that are also Clinal themselves? So their expression is correlated with latitude. And so things at this intersection would be good evidence for adaptive regulatory variation. All right, so starting with the allele frequency Clines. So to do this, allele frequencies were calculated based on the exome capture data for 5 populations along the latitudinal transect. The program latent factor mixed model was used to correlate allele frequencies with latitude while controlling for population structure. So we're looking for a pattern like what you see on the left, where you have, for example, the C allele at high frequency in the north, and then as you go down, you see an increase till the T allele, for example, is at high frequency in the south. So we're looking for that kind of climb. And this can be evidence of adaptation, but again, if demographic processes can also cause this, like isolation by distance. Very lucky for us, there's population structure, but there's not evidence of isolation by distance. So this is a PC plot just showing genetic distance. And as you can see, I'll just show you one example here. This is Pennsylvania, and this is Florida. And so Florida and Pennsylvania are definitely not the most close geographically, but they are clustering together on this plot. So this means that we're not going to get a spurious Clinal signal just due to the population structure here. Okay, so from that scan, we focused on variance with a z-score greater than 2, which is the tails of the distribution. We focused on this is kind of a lax z-score, but we focused on this for identifying overlap with those 2 other signals that I was talking about. And it turns out that most of those things that are over a z-score of 2 are in non-coding regions. All right, so then we looked for EQTLs. So again, EQTLs are loci that explain all or a fraction of variation in gene expression levels. So you can envision a situation where you have this variant G that confers lower expression of gene A than variant C. And so that could be an EQTL. Practically, to identify these cis-EQTLs, what we did was we tested variants that were within 200 kb of a gene. We did things to expression to correct it for covariates that we knew existed, as well as hidden factors. And then we implemented a linear mixed model approach with GEMMA. So here's the results for the cis-EQTL analysis. This is a Manhattan plot, right, where we're looking at chromosome position versus the p-value, and everything above this line is a significant SNP. Reflecting the probe set, the majority of these were falling in gene bodies and in introns. And then finally, we asked whether those were overlapping with genes that also were correlated with latitude, genes whose expression was also correlated with latitude. And so what you're seeing here is another Manhattan plot. And this is a little hard to see, but there are green dots in here. Those green dots are genes with a cis-QTL that also show clinal expression. So those are ones where you identify a variant that contributes to gene expression, and that gene itself is correlated with latitude. And then we also found that of those 132 genes, 17 also were identified as outliers in the Clinal scan. So just to show you that all together, this is a summary of basically everything I just told you, where here we have the selection scan on the outside, the cis-QTL, and then the expression Cline. So we're looking at the overlap between those three different signals, and we're just finding on the outside here have the gene positions of where that overlap is, and there's just 17 genes that fall at that intersection. So that's 17 genes where a variant was a clinal outlier for allele frequencies, that outlier was a cis-QTL, and that gene had clinal expression. Okay, so now that we have those 17 genes, we can then look at whether those 17 genes also have mutant phenotypes in lab mice that are associated with the phenotypes we see along the cline. And then whether those genes were associated with phenotypic variation in wild mice, and specifically body mass, because we had body mass data for all of the individuals that were collected in the field. Of the 17 genes, we found 5 that had phenotypes that overlap the phenotypes of interest from what we see in the wild mice. So that includes things like changes in glucose, triglyceride, adiponectin, leptin, body mass, and activity levels in mutant mice. You'll notice that there are 2 genes, ADAM17 and BCAT2, that are associated also with changes in body mass. And as it turns out, those are genes whose expression is also correlated with body weight in the wild mice, and I'll show you some images of this in a second. And their cis-QTL genotype is also associated with differences in body mass in wild mice. So I'm going to now take you through that for ADAM17 specifically. So ADAM17's expression is associated with latitude, is correlated with latitude. It has a significant cis-QTL. That cis-QTL varies along the East Coast and is a clinal outlier, so it's in a clinal outlier peak. Then when we look at ADAM17 expression versus body mass, and this is actually body mass that's already been corrected for latitude so that that doesn't cause a spurious association, we see that it's significantly associated. The expression of ADAM17 is significantly associated with body mass. This is also true in individual populations, so this is from all of our individuals, but we also see this in just single populations, which indicates this is not an artifact of population structure. And then finally, so that cis-QTL variant that's associated with the expression of ADAM17 and is varying clinally is also associated with body mass variation. And finally, we were also able to validate the ADAM cis-QTL in European individuals. So we downloaded some data from Har et al. 2016 and found that this variant associated with the expression of ADAM17 is also associated with the expression of ADAM17 in European individuals. It's a similar story for BCAT2, where we see that this gene is again the expression of this gene is associated with latitude. It has a significant cis-QTL. Body mass is associated with expression of BCAT2, and we have a significant cis-QTL that's associated with body mass. So like I said, these genes are associated with changes in body weight in lab mouse mutants. They've also been implicated in human metabolic disease. BCAT2 in particular, the expression of it has been associated with different transcription profiles for obese and lean humans. And ADAM17, polymorphisms in ADAM17 have been associated with obesity risk as well as metabolic inflammation. Okay, and so to sum that up, at the intersections, we were able to identify 17 genes that were associated at that original intersection of things that had an EQTL, showed clinal expression, and that EQTL varied clinally. Then we were also able to identify 5 genes that had mutant phenotypes that were relevant to the phenotypes that we saw in the wild mice. And then we were able to identify 2 genes who we could directly associate with body mass through expression and the association between their EQTL and body mass in wild mice. All right, so to sum up part 2, I started by asking what the role of regulatory variation is in environmental adaptation in house mice. We found evidence for a lot of regulatory variation in these populations. There's evidence for adaptive cis-regulatory variation along this latitudinal cline, and we've also identified candidates associated with adaptive phenotypic variation in body mass. Okay, and so then I really quickly, before I wrap up, want to tell you that this is not, that we've looked at other aspects of this as well, including looking at copy number variation on the East Coast. So copy number variation is where you see amplifications or deletions relative to a reference genome. And we've found that there's there's quite a few copy number variants segregating on the East Coast of North America in these mice. So here I'm showing you red lines equal a gain relative to a reference, blue equals a loss, and then lines that are kind of purple are is where we see a loss or a gain depending on the individual. And we then looked for whether copy number variation was also associated with latitude. So do we see a situation where copy number is high in New York and then it's much lower in Florida, for example? And so we performed a genome scan for that and found 2 regions where copy number variation seemed to be clinal. What interesting thing came out of this is that in one of these regions we have this gene TRPM8. And this is a gene that's recently been associated with adaptation in other systems to cold. TRPM8 is actually the primary molecular transductor of cold sensation and plays an essential role in physiological thermoregulation. It's also been implicated in adaptation to cold in ground squirrels and Siberian hamsters, potentially humans, as well as woolly mammoths. So it's a really interesting gene to follow up on. Okay, so bringing this back to my very first slide, I hope I've demonstrated to you with these 2 projects that, at least in house mice, there's strong evidence for an important role of regulatory variation in adaptation and speciation. Okay, I really quickly want to thank a bunch of people. Obviously, I'd like to thank Michael and my committee and other people who've given me feedback or helped with various aspects of this project, including Noah Whiteman, Noel, Lydia Smith, Lelania, KB, as well as all of my co-authors. I want to thank a lot of my lab mates and the Nachman Lab, who've been a really great environment to be in for the last 5 years. For the last 5 years, have gone to Thanksgiving with all of my lab mates, and they all came to my wedding. So I couldn't have asked for a better group of people to be around for the last several years. And then finally, I also wanted to thank the IB and MBD community. It's been an amazing privilege to be here, and that's something I feel most keenly. So thank you for listening, and I'm happy to take any questions.
All right, we got time for questions.
You might have said it and I missed it, but was the reason why you picked liver for the gene expression analysis you're doing?
Yeah, because it's a very metabolically important tissue. That's pretty much what it shows it, yeah. It would obviously be interesting to look in other tissues, but we didn't collect those in the field.
I was trying to think about the results about the Cis-men trans effects. I was thinking potentially you could have a situation where you have a evolution of a transacting factor upstream of a whole bunch of downstream genes, I'm thinking about sterility here, and that could then, because it's messed up one particular gene that's upstream of the others, that could then affect all the chain going down.
Yes.
That would kind of appear like trans, I believe. Or, I'm just wondering, can you decouple that sort of scenario from multiple independent evolutions in multiple genes?
When we say that there's widespread misexpression in a species, you definitely can't decouple that from like there's widespread misexpression maybe just because one transcription factor got messed up and did everything. But in the case where we were specifically looking at individual genes that have a cis-regulatory variant and a trans-regulatory variant, the fact that those are enriched for misexpressed genes suggests that that's not the case.
Other questions?
All right, so in the little teaser that you had there at the end about the copy number variation in that one particular gene, is there lots of copy number variation in that in lots of mammals, or is it just something that you seem to be finding in plus?
Yeah, I have I've looked around and there doesn't seem to be a lot of evidence for copy number variation in other animals at that gene, yeah. Most of, so I mentioned there's a couple studies that have identified that as important to, cold regulation, and those are mostly like protein coding changes. Yeah.
You looked at some of the alleles that you identified as interesting for adaptation in European populations. And do you know enough about the history of colonization of North America and sort of what the genetic stock is for these mice to do other kinds of comparisons like that?
We assume they came from Europe. We don't know anything, and we don't know specifically where they came from. We'd have to have better sampling in Europe to say anything about that.
And are they just domesticus, or are they?
Oh yeah, the mice that I was looking at for Europe are domesticus, yes.
Sort of a related question, do you think these adaptive variants are new mutations in the Americas, or are they segregating polymorphisms?
No, I think they're segregating polymorphisms, and I think that because there hasn't been a lot of time, and the fact that we were able to validate the Cis-regulatory for ADAM17 in Europe suggests that these are standing variation.
Have mus, domesticus, and musculus invaded other continents, and do they do the same thing there that they've done here?
Yes, they've invaded other continents. I don't know, do they show Clines in South America in body size?
Yes, not as strikingly as in North America.
Yeah.
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 And they've invaded Australia, but I don't know of any documented Clines and bodies. I don't know that people have looked.
Yeah. They're capable of getting everywhere. Yeah.
Could you get any of the changes that you found along the Cline? How long does it happen for mice to adapt to the different temperatures?
So they've only been there for 300 to 400 years, but presumably, I think this is all from standing genetic variation, so.
I see.
Do you see any chromosomal biases where your regulatory variants are? Or like, is it some more on the X chromosome or autosome?
We didn't look at the X chromosome, unfortunately. We just looked at autosomes.
Okay.
Yeah, so I'm not sure. Oh, yeah.
Hunter Fraser published a note recently saying that the appearance of compensatory evolution in cis-trans variants could be due to just the way that they're estimated with F1 hybrid data.
Yeah.
Is that something you've thought about with your data, or that you're planning on looking at in your postdoc?
Yeah, well, yeah, he's actually taken our data and is looking at that now, so we'll know soon.
Are you guys doing like the whole replicate hybrid approach? Or
Yeah,
something?
yeah.
Okay.
Yes.
So if I understood it correctly, at the end of the first section, you indicated that the misexpressed genes were concentrated within these reproductive. Was that the case, or was that?
No,
Or
I
were
mean,
you
they-
just emphasizing this?
I was emphasizing that a lot of them have reproductive phenotypes. There's not an enrichment for reproductive phenotypes.
Okay,
Yeah.
I guess my question is, would you expect that there would be a lot of, that most of the misexpressed genes would actually have some connection to reproduction?
Just because we were looking in the testes,
Okay.
yes.
think it was Professor is looking at these just tremendously large rodents, and they were house mice, right? Which
Yes,
is like
yes.
grotesquely large.
Yeah,
Okay.
they're domestic.
So, have you had a chance to look at those data and see if some of the same sort of Cis and QTL show up there? I mean, is it everything turned up to the max in terms of EQTL?
Yeah, they
Produced
haven't
that?
looked at eQTL yet. They've done QTL mapping. I think they're planning on looking at eQTL mapping. It'd be interesting to see if we see some overlap there, but I'm of the inclination that there's a lot of ways to make a fat mouse. And so I'd be a little surprised if you saw a good overlap between what we're seeing here.
So sort of related to that, Katya, I was surprised you didn't say anything about effect size. So
Oh,
in
yeah.
humans, there are over 100 genes that have been identified that affect body mass, and collectively they account for less than 3% of the variation in body mass index in humans. But you know, what's going on here, and you know, what can you say about the effect size of genes and the likelihood that in different populations that have evolved under the same selection pressure, there might be similar or different responses?
Yeah, so the effect size of these variants in comparison to what you see in humans is very large. So these explain together nearly 9% of the variation in body mass. And that's a very rough estimate given our sample size. But that's a lot bigger, and that's likely because humans are probably under stabilizing selection. Mice are not.
Okay, any more questions? I'll remind everyone that there will be cake here at 3 if you want to come and join Katya in celebrating. Thanks very much.